after the first two speakers, I dare to be a bit provocative as well. I was not to do that at all, of course. As people know me, I will never be provocative normally. But you already see my title, The Stupid Biologist View. Actually, I know Giancarlo via Luis, the big guy there in the corner. And I never realized that there was any connection between our worlds until the first speaker, when you mentioned Barry Smith. Because we have a term in biology called the Barry Smith syndrome, which is very clearly defined. For example, I remember one day in Dagstoel in the wine cellar with Barry and Mark Muse and another. We spent, I think, 4 hours to discuss whether an absent nipple, which is a genetic defect where you miss one nipple, whether an absent nipple is a nipple or not by virtue of being absent. Drove me completely crazy. And it's the same thing like you have the concept wind speed, and then the wind speed can be 0. Is that still wind? No human would ever care about it. You all know what we mean. There is no wind when the wind speed is 0. So the thing is that I think what Enrico just said, you guys try to be so precise because you deal with machines who are entirely stupid. And humans are not precise. If I love piano music, I don't care whether you think about a grand piano and I think about a small piano, because it doesn't really matter. So I'm most interested in the concept knowledge and qua that I discussed a lot with Giancarlo at the time. And you remember the last paper he's trying to get published, I think, still on ontological unpacking. So I was reviewing it, and just as a predicate on what I'm going to show, I wrote this little note, probably on 20 places in the paper, you remember. And you said, what do you mean here? So it's all these things only come up because you want to be so precise that even machines get it, And this is more or less what both of you said. But anyway, so let me give you the stupid biologist view. And I was not going to say anything about ChatGPT, but now I cannot avoid it. So when I did my inaugural speech a long time ago, I'm a silverback, the title was Knowledge is like love. It multiplies when shared. Okay, that was just to have a fun title. And it was there for years, of course. And then ChatGPT came, and the first version, not Giancarlo, somebody else, typed in, what's the major contribution of Barend Mons to science? And it says he's a major expert in love. He has really changed love. So if you want any advice on love, come to me. It's totally crazy. So in the last version of ChatGPT, Giancarlo and I were joking on when we had a long discussion online, and it says I was a co-founder of the gene ontology. Hallucinating again. And we decided, it's a theory, that if we would have written Go Fair, which I'm the director and it's always associated with my name, with a dash in between Go and Fair, this would not have happened because the stupid computer sees GEO, which is the acronym for the gene ontology. I'm a geneticist, lots of papers about genetics, so it hallucinates that I must be a co-founder of the gene ontology. So what I really want to do here is to see how we implement logics or ontology or whatever. Now we just heard we don't even know what implementation is, at least in your community. Well, I do. At least some form of interpretation. So let's go there. So the reason that I actually know Giancarlo is via Luis, and it was this meeting in 2014. We will soon have this 10-year celebration, where we defined the FAIR principles. It went way over 10,000 citations. Nice. Doesn't matter anything to me. I always say half of the people that cite the paper have clearly never read it. And then Luis always says half the authors also didn't. So, but it has a high impact for sure. I'm not going through the principles, but it's definitely not a standard. But just show that red principles are essentially technology, the sort of things that computer scientists should do right. But there's also a lot of principles that really require a social contract. So what is a persistent identifier? How long do we want it to be persistent? Does it resolve only to the intended defined meaning, whatever you use as a term there? How do we decide how rich metadata should be for other people to be able to reuse my data? And So that's all I want to say about the FAIR principles, but I believe that if you make a conceptual model, it should at least be also fair so that machines and people understand it. But people are much more able to deal with what I call near sameness. Okay, so when I listen to you two, I created a knowledge, I'll explain later, in my mind. So now I have my opinions about you and my knowledge about you. And every time I hear your name, I have all these associations together. Machines, unless they have knowledge as well, can't do that. So a little bit before I go into implementation about, what is misperceptions about FAIR. The first one is that the next standard, and you all know the story that you have too many standards, so let's make another one to supersede all the standards, and then you have one more. No, it's a set of rules to make sure your stuff is machine actionable, not just machine readable, but machine actionable. That's extremely important for biology. Why? When I plug any machine in the wall now, it starts spitting out terabytes of data. People can just not deal with it. And there is one more reason why it's so important to connect biology and your field. That's, I think, why I was invited. Because when Luis came to my group, he's a stupid computer biologist from Twente, you need to learn at least a little bit about biology before you can function in this group. So I gave him a crash course on DNA, RNA, protein, how information is handled in life, with an incredible amount of imprecision and redundancy. Otherwise, life would never exist. You would just die out immediately if you're precise. So after that crash course, Luis made a historic remark. He said it should be forbidden to study computer biology before having the basics of biology. We did everything wrong. Okay, that's maybe said too much. So the other misperception about FAIR is that it is open by default. You can have very sensitive data and make them fair, and then the A is very strong. It's not accessible by anyone else or any computer that is not authorized. It's not just for people. A lot of people just now say that they go FAIR, but they totally neglect the idea that FAIR intrinsically means machine actionable. So they say, oh, I put a DOI on my PDF, now I'm FAIR. No, not really. And also, it's not for all data. So we say all these legacy data, only make them fair the first time you want to feed them to a computer. Otherwise, don't do that. Maybe fair metadata. A lot of the principles are about metadata. So, and that is anyway disappearing in the rear mirror in my field because we double our data every 6 months. So the log phase of data. In 10 years' time, the data we have today are close to zero compared to what we will have then. We will not be able to store it on hard disks. We'll store it either in DNA or something even more clever. The whole internet can be in 1 kilogram of DNA, and all the genomes of all people in the world can be in my little finger because I have 8 billion cells, and they all have my genome, which is 2 DVDs per cell. But it's all here, including the readout system. So biology has done a few things that you probably can learn from. That's the point here. Now, to get good data stewardship, we need to spend about 5 to 10% of any research grant in industry and in the public sector on FAIR and good data stewardship, which is one of my most hated papers. The most hated one is "Which gene did you mean?" Which has the statement, why text mining, why bury it first and then mine it again? So we bury everything in ambiguous text, and then machines have no clue what we mean. We have to do text mining to create triples again. But really, if we want to do the science of the future with enormous data sets, we need computer-assisted reasoning. You have another term for it that was basically the same thing, augmented whatever. The reproducibility crisis is partly due to the fact that stuff is not machine readable, and therefore people that work in data science waste 80% of their time data munging before they can even run their algorithm and have a clue what the data means. So far the complaining. This is my key slide, and I'm afraid that I will be really challenged by this community, but that's fine. It's a somewhat different day today from normal. So another guy that I worked strongly with is Eric Schultes. He came to Leiden about 12 years ago. And he works on complexity theory, spent some time at Santa Fe and so on. And he came up with this sawtooth graph that you see in many physical processes. You know, the forest fire model, you put more and more trees, the whole forest doesn't burn down. You put one more tree, the whole forest burns down, which is with this percolation point. And my challenge to him was, can you also see this in how we discover knowledge? So he made a very simple model with Herman van Hagen, one of my students, and they put increasing complex patterns on the x-axis in the model. And here they put the number of CPUs or units of intelligence that you need to understand the pattern. You could say to predict correctly the next step in the series. Now, it's logical that if you keep adding intelligence or CPUs or whatever you want to call it, and stuff gets much more complex, you will still see all the patterns and be able to recognize them. However, if your intelligence at some point gets limited, like for humans, we cannot put all our brains together. We can put a lot of computers together, but still there is a limited number of CPUs or units of understanding. If things get a little bit more complex, your understanding goes asymptotically to zero. So you get the famous sawtooth, even in knowledge discovery. And what we said is, if you go ontological, you describe what you think you know. You still have things like, of course, you know that sentence, the old man, the boat. We all understand that it's still a sentence. The old people man the boat, you know, but for ChatGPT would be a pretty nice challenge. So if you model what you already know, you can probably use static graphs or ontologies. But if you go to knowledge discovery, you don't know what you're talking about. And many major discoveries from the first, maybe we turn around the sun instead or the other way around, on the stake, you know. Many of the major discoveries start as heresy. So at some point, we go down to zero. First interesting idea is that if you would be able to continue to add intelligence, we go to the very end. There is no chaos, because you will always see patterns and understand stuff. And we call that the global online database because it's a nice acronym. Yeah. So God. If you, however, go into biology now, one project I'm involved in is the Human Immunome Project, where you want exposome, epigenome, microbiome, all these things down to the genome, each terabytes of data, by the way, per person. And we want to understand the entire human immunome and why African people react differently to a vaccine. And this could be environment, this could be genetics, it could be transcriptomics and everything. My guess is that this is way beyond human understanding. The actual process we want to study. Probably humans understand this little bit. But everything out there, which is maybe hallucination of ChatGPT, bears can make their own rockets to fly to the moon. Well, maybe in 100 years it's possible. And you're a famous example, Giancarlo, of a surgeon can transplant his own liver. In 10 years it may be true. So there is a lot out here that we perceive as chaos because we have a lesser mind. And this line, we showed also retrospectively, moves to the right. So we get more and more knowledge, therefore we understand more and more complexity, and therefore we have also more and more compute power. And that's, I think, very important of your work, that you make computers understand. So a few words about nanopublications, and then my major plea for dynagraphs, and I tried this on a knowledge graph conference at Daagstoel, and it was sent out. I wasn't allowed in the wine cellar anymore, so maybe I hope you were less aggressive. But the idea here is that if you make a single truth statement, let's call it a very simple thing, it does not even necessarily true. That's the whole truth maker, truth bearer thing that we are now discussing. Let's say you say hydroxychloroquine treats COVID. Published in the Lancet. Not the worst journal. So you need provenance, and a unique identifier for the assertion, that it was made at a certain point, regardless whether it was true or not. Now we don't believe it anymore. Well, there's two people still believing it, Trump and Bolsonaro, but that's it. So we want to be able to say, okay, this statement, the same subject, predicate, object, each with their own globally unique identifier, was made 10,000s of times. Okay? Every time the provenance is different. Who said it, when, where? If we then introduce the concept of a cardinal assertion, that means we store this triple only once. Okay? And we have 10,000 provenances by now, including it was published in the Lancet and then contested by 800 orchids that don't believe it anymore. Even machines would be able to discard this triple as probably not true. Okay? Or not relevant anymore. But the fact that it was stated is not changing. So non-application should never be changed. There's no versioning or anything. There's snapshots. Cardinal assertions change not the assertion, but their provenance files that are separate change all the time. And then what we do is we call this noded. Now this could be any of the previous speakers, and all these assertions about you, Italian, form in my head. So I have this whole noded, or fair digital twin as we call it now, about you. Now these are all the assertions we have made, for example, about a disease or a drug or a person or a country, whatever, any concept. Now, to give you a few numbers, how to deal with our amount of information, not in knowledge graphs, drive me crazy. The idea is that we have about 10 to the 14th nanopublications in the life science. There's no computer dealing with that efficiently today. We have about 10 to the 11th cardinal assertions. That's already 1,000 times reductions. That simply means the average assertion is out there 10,000 times. Malaria is transmitted by mosquitoes, 100,000 times. So on average it's 1,000. So you have 10 to the 11th cardinal assertions. In the entire Medline database, 40 million articles, we only publish about 1 million concepts. So we have only 1 million nodes. And they form a dynagraph that we deliberately do not connect these nodes. They float free in the chaos of this graph. And some of them we experience as Brownian movements. Sometimes the nodes become so close in their inner product in multidimensional conceptual space that even the computer says, hey, maybe this gene is involved in this disease. So we predicted 140,000 new gene-disease associations. And in 10 years' time, people have only found 6,000. And the reviewers of Nature Genetics kept rejecting the paper. And in the end, the only argument was, we cannot handle this. Well, that's fine. But the machine saw all these patterns out of 240 million possibilities. We still have these possibilities here. So better study them. So why do we need fair data and your type of work? So I don't dismiss it at all. Here you see a type of a schematic representation of a nanopublication. Of course, there is RDF behind this. You don't want to see that. This is a variant in the genome. And here it says exactly where it starts and ends. So that's the nucleotide position, which is the reference genome and chromosome 1. So essentially, this describes exactly where the mutation is in the genome. Okay? If you go to the Leiden Open Variation database, where doctors from all over the world put these statements, oh, I found this strange variant mutation in the genome of my patient, and he has this disease, maybe there is an association. Maybe. Then we worked also on another paper which is published in Nature, the Twente 5, and we made nanopublications, not we, but people in my group who are on the paper. And this is where the transcription start sites are. Forget it. Transcription start sites for people that are non-biologist is almost, if you start there, the transcription of the whole gene plus the upstream sequences is the README file in your terminology. So it tells you, make this string of amino acids, then you have to fold it and cut it. So it gives instructions for what we call post-translational modification. There's 94,000 of those in the genome. There are 25,000 genes, or less. We have 95,000 transcription start sites, but don't forget it. In the 98% of our DNA that we used to call junk DNA, maybe you heard the term. Of course, it is not junk. Everybody knew that. But we never knew what it was. Until recently, we found that there is 4 million of these regulatory sequences in this 98% of our DNA. And only 5 or less percent of the DNA is coding for protein. So my question was in those days when we started to find out, and I explained it now as a pianola with 25,000 keys, and the roll has 4 million pins, so you can play a fruit fly or a human. It's the same genome, no problem. That's the melody. If there is a mutation in the gene, the protein is not made, you're almost dead if it is an important gene. But what if a lot of genetic diseases would not be in the genes, where we have been looking for 20 years, but could be in the transcription start set? Some pins are wrong, you play a little bit wrong melody, but it is still Bach or Father Jacob if it is a bacterium. So there is also no correlation between the complexity of the genome and evolution. You know, a cucumber has 4 times more genes than you have, or at least than I have. So now imagine that you want to ask the question in biology, talking about implementation, how many of these 2 million variants that some doctors have reported as potentially associated with the disease in LOVD, the Leiden Variation Database, actually map to a transcription start site, or to a promoter, or to any of the other things. So anything outside the genes that really changes your behavior. And again, this is part of this redundancy that life doesn't care about precision at all. Then we would all be dead, or never exist, actually. So this question is not even posable logically unless you have 80,000 PhD students to read 40 million papers. So the idea is now that we have 2 million or 94,000 nanopublications describing exactly where transcription start site is. They have been made to completely separate from the LOVD ones. These 2 groups have never met, but they properly construct their nanopublications. And now what you can see very easily is that the machine is able to say, okay, look, here's a start and end site, there's a start and end site. So it concludes that this transcription start site is disrupted by this mutation. So in 5 minutes, we had the answer. And the answer was that of the 2 million variants that have been associated with disease, almost 20,000 were mapping to a transcription start site. So they disrupted the transcription start site, you made the wrong protein in the wrong tissue, you get sick. There's even a guy with 4 feet. And we know for sure, although it's not proven yet, that the transcription start site that normally sets up the whole cascade to make a hand is broken. So the enzyme takes the next one and makes a foot. Because all the genes creating a hand or a foot are identical. Look at chimpanzees, you hardly see a difference between their hand and their feet. So, and there is only 2% or less difference between the genes of chimpanzees and us. I worked with chimpanzees for years, and I'm sure that 90% of the huge difference between chimpanzees and us, including that they're 10 times stronger, is not in the genes at all. It's in the regulatory sequences. Well, anyway, this is the sort of complexity we're dealing with. That's why we need logics, nanopublications, ontologies, but an ontology that tries to be so precise. That's the whole discussion after Giancarlo on his paper. That's why I put the noted everywhere. If we accept mere sameness, like life does, and like how we think, you know, then suddenly we understand each other, even if we have not exact agreement. If you try to go down to the exact definition of everything, you run into all the Barend Mons syndrome. I will find a new name. So this is just 2 more examples of how we implement this now to see that your stuff is actually implemented in biology. But please learn also from us. This is a disease model. We have a graph of all these 10 to the 14th nanopublications collapsed in 10 to the 11 assertions, the cardinal assertions in a graph. It's a commercial company called Euritos. And within an hour, you can make a model like this on how is the disease progression in COVID from being a bit flu-like, nausea, blah, 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 to vasculitis. Then your vessels start to leak, then you get thrombosis and your lungs become 4 times as heavy as normal, and you die. Cessation of life is a euphemism for that. The orange balls are all the genes involved. And then what we can do now, we take and top all the drugs that people have thrown into this in panic to see whether they could treat COVID. One of them is hydroxychloroquine. And as you can see, there is one line to one gene in the whole disease model, which is also associated with arrhythmia of the heart. So in the very early days, we already said, computer says no, in a way. This is very unlikely that this drug works for COVID, and it was proven true. And of course, there was a rush on hydroxychloroquine, so we couldn't use it for malaria anymore. It was even worse. Then you see the green ones, losartan, tocilizumab, dexamethasone, all have these bundles of connections in the system. So they were highly predicted by the machine as being potentially useful for COVID. And all of them are still in use. So you can see that you can do very much predictive stuff, almost like a petri dish or for an in silico study. And if you then dig down, and here the conceptual modeling comes in, I see the conceptual model and the constraints that you put on your graph, which could also be the solution for the crazy stuff that comes out of the bars and the JetGPTs at the moment, by having constraints. You cannot make a protein cannot make a protein-protein complex with a building. So you can rule out a number of things that are completely illogical. So you constrain the computer. But the beautiful thing is here that when you combine this with wet lab experiments, so we have organ on a chip, where you have little pieces of organ, so lung tissue, intact vessels. We pumped, how do you say this for non-biology? You perfuse serum of very sick patients that were almost dying. And what you saw, indeed, that prostaglandin E, which is something dexamethasone inhibits, and dexamethasone happens to be positive for patients, causes vascular leakage. If it's overproduced, you get holes in your vessels. Well, you can imagine if that combines with thrombosis, blood clots in your lungs, you're dead. But when you pump this stuff through intact vessels of lung tissue, when the thing comes out, the serum, there's even more prostaglandin E than when you get in. There are intact endothelial cells at the inside of the vessel. So they produce prostaglandin like crazy by something that is in the serum that was induced by the virus. And that happens now to be thromboxane B2, forget all these names. It's a tiny fatty acid that we are now trying to inhibit. So then you go back to the knowledge graph and you say, where does this fit? And you see everything makes sense in this graph. But if you want to make this graph by reading papers, forget it. So on the one hand, yes, notifications, cardinal search, and knowledge are extremely important, but treating dexamethasone and its knowledge with 10,000 triples in it as a free-floating molecule or a fair digital twin of the actual drug is much more useful for knowledge discovery than precisely putting it in an ontology. That's dead. It's solid knowledge that we knew already. So maybe the precision versus, vagueness and near sameness thing is really when it comes to knowledge discovery. Just 2 more slides, then I'll finish in time. We recently got another question saying, hey, all these people that are incredibly overweight, well, this came from the United States, but okay, they have frequently diabetes. Now there is this tendency, suddenly, that a lot of these people are prone to early onset Alzheimer's. Imagine the epidemic that could cause in the United States, also increasingly here. Question was, which are the genes involved? If we do very much the restriction that we have, constraints on them all, we still end up with 68 genes to study. Each of them has hundreds of papers about it. So, this is the sort of complexity we're dealing with. But we can now zoom in and make more constraints, and in the end, you get a manageable number of genes to at least focus on as a potential cause for this connection. Last slide. We actually have a project just awarded called Staying Ahead of the Virus. And this is the spike protein that we all have heard about, part of the vaccine, or Pfizer, for example. So we have a fair digital twin of the protein, including every amino acid position, what the function is, all these things are there. And if you allow 3 amino acids to mutate, which is very likely to happen in the coming 10 years for each of those proteins, you already get for one little protein, it's a small protein, 1274 amino acids, 230 billion variants. Okay? They will come up in the coming 10 years. Luckily, we can show that 90% of them cannot even exist physically in the Goldilocks environment. They will just fall apart because they are wrongly constructed. But some of them will get rid of the antibody binding sites, so they may escape the vaccine, or they bind much better to the cell, so they are more virulent, like Omicron was. It luckily was not so sick-making, so it was the perfect live attenuated vaccine in a way. It saved them, I think. Now, we want to know why certain people react better to the virus than others. Those kind of things are in HIP. But the interesting application and implementation of this sort of technology, and your work is fundamental, literally, to that, like you can basically see here, is that if we test all these variants that may come up very soon, we test them indeed for escape of the vaccine and so on, and we end up with 1,000 or even 100,000. Pfizer told us they can easily construct the RNA of all of them and have the vaccine ready whenever that variant would appear at any airport. In 100 days, they can have enough vaccine to vaccinate the world. That's the sort of applications we do. Again, finalizing, your work is absolutely fundamental, but my plea is let go of this precision. Thank you.